I want to begin by introducing my colleague, Winona Verketer, who's back there. 21 years ago or thereabouts, Winona got her PhD in our department here at UC Santa Cruz. She then went on to NASA Ames for the rest of her career to date. And we have gotten together 20 years after she graduated because you're going to hear about a collaboration that we are undertaking. By the way, also 20 years ago, Winona and I flew down to JPL to give a talk about how nanopores could be used as life detectors. It fell on deaf ears because nobody knew what a nanopore was. We had invented the term here at UC Santa Cruz in a paper just the year before. So here we are now 20 years later, and I'm going to tell you what a nanopore is. But first, I want to say that Steve and Sherry challenged us to challenge the audience. That was the instructions we had. So I thought a good way to do that is to ask 5 questions right at the start that are related to what I'm going to talk about and answer them in the summary. So think about what molecular species are unique to living cells. And of course, just think about that. Don't answer out loud yet. Can we detect such species as single molecules? If we can, and if it is unique to life, that detection should say, yes, there's something alive on Enceladus or Mars or Europa if we see this signal that I'm going to show you. The nanopore signals you'll see. So how about the cost of doing this? I forgot to bring it up here, but I did bring a little handheld device. It cost me $3,000. And with that, I can see single molecules of DNA go through a nanoscopic pore. That's how easy it is these days to see single molecules. Under what conditions can they persist for thousands, perhaps millions of years? You may know that we are extracting Neanderthal DNA from some several tens of thousands of years ago. And if you go visit Jurassic Park, the movie, you know, they're getting DNA from dinosaurs, you know, several hundred million years ago. Well, that's not quite right, but certainly millions of years it's likely if it is frozen in ice. Finally, are such molecules valid biosignatures? And I'll be answering those questions. So what is a nanopore? Here's the first one we worked with. It's called hemolysin. It's a bacterial toxin released by Staphylococcus. It punches holes in membranes, and that's why it is a toxin. And you can see that little sort of a mushroom-shaped structure on the left. That's an X-ray diffraction. It's the real thing. That's what this looks like. And you can see the stem poking through the membrane. And off to the right, you can see a single strand of a nucleic acid just barely squeezing through that. So we knew that this might be a channel that we could use as a way to watch single molecules go through. So hemolysin is a heptamer. If you count those colored subunits, they self-assemble into the membrane to make a pore about 1.5 nanometers, 15 angstroms in diameter. So if we look at a molecular dynamics simulation of DNA going through the pore done by Alexei Aksimentiev at University of Illinois, this is what it looks like, about slowed down by perhaps a millionfold. So a voltage is imposed across the pore, and this is basically single molecule electrophoresis. And you can imagine that if we have some way to sense each of those bases going through a limiting aperture, we might be able to sequence the DNA as it squeezes through the nanopore, and that was our idea. So I carried some RNA back to NIST, where John Kasyanowicz had a hemolysin pore up and running. and he expressed interest in the possibility that we might be able to see it go through the pore. So if you look off to the left, you'll see where we added the polyuridylic acid. That's a synthetic DNA composed of UMP subunits. And you can see that as soon as we add it, we begin to see these downward deflections. And those are blockades of the ionic current. We know that it was poly-U doing this because we added an RNase that would break this up into little pieces. And suddenly we see many more blockades, which slowly get shorter and shorter and finally disappear. That was a strong clue that we might be on the right track for nanopore sequencing. Now then, I want to show you how fast they go through. And once again, there is an ionic current through the open channel, which is blocked when a piece of DNA or RNA goes through by the applied voltage, plus and minus. So watch how fast this goes through. So that's what an oscilloscope screen looks like. And every one of those downward deflections is one molecule being pulled through the hemolysin nanopore. So we published that back in 1996, and since then there's been quite a bit of progress. This is the next publication, 1989, all done here at UC Santa Cruz, by the way. Mark Akeson had joined my lab. And if you notice now, we can see the entire blockade. Here it enters the pore, goes down, but instead of being flat at the bottom, there's two levels in each of those blockades. And the reason is that it is 100 mer of RNA going through, composed of 30 adenines in a row and 70 cytosines, and a little GMP at the end. So this really helped out on our first patent, because we could now claim that we were seeing the portions, the sequence of bases in these strands of RNA. So the patent issued in 1998, and a company called Oxford Nanopore licensed it. The reason is that where they got their name, by the way, is right here. Watch a square appear down below. Here it comes. We invented that word to describe the pore that we were using here. So a nanopore is where this word came from in this paper. So Winona started her PhD work in 1996, finished up in about year 2000, and here's one of her papers. The reason that I show you this is that there is a lot of skepticism about what we were seeing, whether we would ever get this to work. So Winona and we, thinking together, had an idea that we would put in a hairpin of DNA. So right there is a hairpin piece of DNA where you have a double strand portion in the stem going up to a loop, and you can see why they'd be called a hairpin. We predicted that we might see it caught in the pore and stuck there until it could unzip and go on through the pore. And this is what we saw. Here's a typical signal of a 6-base pair DNA hairpin. Here's the open channel current, 120 picoamps. Here, that molecule, single molecule, is caught in the vestibule of the pore. Here it is sort of jiggling around, and suddenly it unzips and comes out the other side. This is what we saw when we varied the number of bases in that hairpin. And you could probably predict, just from what I showed you, that a 3-base pair hairpin would be relatively unstable and would go through unzipping in a millisecond, 10 to the 0, 1 millisecond. So here's a bunch of those. And then as we added base pairs, it got longer and longer, and out here it was many seconds for an 8-base pair to unwind. The most interesting thing is that we could predict from thermodynamics how long these would stay in the pore, because we had the stabilizing energy of the hairpin. So that attracted interest of places like this startup company in the UK called Oxford Nanopore. They began to license our patents. This sort of put it over the top in 2012. This is our first sequence done by Nanopore Sequencing. We got together with Jens Gundlach at the University of Washington, who had discovered a better pore to use, and we had found an enzyme to slow down. So instead of going through at 2 microseconds per base, it is now advancing through on the millisecond time scale. Adenine, cytosine, thymine. Adenine, cytosine, thymine. Adenine, cytosine, thymine. We were watching a single molecule being modulating the current as it was pulled through the pore, and is being controlled by an enzyme that was actually polymerizing DNA from a single strand into a double strand, is pulling it through the pore. So Oxford got busy. We discovered that Oxford and a business can do things that we just couldn't imagine. We worked for 10 years with 1 nanopore in a lipid bilayer. Oxford came out in 2012 with 2,000 nanopores in a little sensor array, each one of them individually addressable. So that was quite an extraordinary thing. As part of their advertising, in a sense, they said, let's send a MinION, one of these nanopore sequencers, to see if it works in microgravity. So Kate Rubin, floating in the International Space Station, used a MinION to sequence DNA. Here it is, the signal's coming from there, and here's the DNA signal coming out onto the computer screen. Well, that is the story of nanopore sequencing, but now I want to finish up by telling you about nanopores. We can now drill, one last slide here. These are all for sale now. The company had a stock offering. They brought in $6 billion, and they're now leading the way for sequencing human DNA. This guy is called the Prometheon. It sequenced a human genome in 5 hours up at Stanford. One of the patients there having a genetic disease that was diagnosed by that readout. So that's what we're doing. The first human genome took 10 years and $3 billion. We can now do it for under $1,000 in 5 hours. So we can now drill a hole, a nanopore, in a solid state. And if we're going to send a nanopore off to the surface of Mars or to Enceladus or the surface of Europa, it's got to be pretty stable. These are much more stable than protein nanopores. And here's one that there's a 5 nanometer scale. This is about 7 nanometers across. And we can drill them down to about 2 nanometers, which is the size of the hemolysin pore now. So a company here in town called Antera, startup company, built this device. It's called a Nano Counter. This is a little chip that you pull out, you put your DNA sample into the Nano Counter, push it back in, and right away it begins to read DNA that has been deposited on that chip. And this is what it looks like. Here are single molecules of, in this case, RNA. Let me get this up on slide. So this is ribosomal RNA, and each arrow is a single blockade of a molecule going through the channel. And this is what that blockade looks like. Here's the open channel current. Here is a piece of RNA going through and out the other side. Here's another one lasting a little bit longer. That's this one right there. So we can see single molecules of nucleic acid with this little device that cost me $3,000. It's really inexpensive to be able to do this sort of thing. You can also do DNA. So here's a DNA from a bacteriophage with 48,000 base pairs. And each of those is one of these molecules going through the nanopore. Here it is cut in half again, cut in half again. Here we are down in about 800 or so. think that's what that is. So this is really good technology. It's inexpensive, and my point as I finish up here is that if we saw something like that on some melted ice on the surface of Mars that we drilled down for, or some melted ice on the Europa surface, if we saw blockades like that and points like that, I would be pretty convinced myself that life existed there. So how to detect biosignatures on other planets? They have to be polyanions, because they have to be pulled through the channel by a voltage. The ice sample would be melted. We have to add a electrolyte to carry the ionic current. Microfluidic delivery of that melted sample to a 10 nanometer solid state nanopore. We then measure the ionic current going through. We look for transient blockades like I showed you. And if we see transient blockades, I think they would be an polyanion. And the only source of polyanions that we know of are living organisms and the backbone of DNA and RNA. So that is what I have to tell you about. Blockades are strong evidence of microbial life. If we have interested any of you, Winona and I are putting together a proposal to go to the next step with this. And what I would like to invite you to do is, anybody is interested in this, please do contact us. We'd be very happy to talk to you about what this work is all about. So the answers to those questions: yes, polyanions such as nucleic acids are really unique to living cells. Yes, we can detect them as single molecules. I've shown you that. Yes, we can do it with solid state nanopores that are very stable. They can last for many, many years if they're frozen in ice. And finally, I think that that would be a valid biosignature. Thank you. Okay.